Welcome to Track 4, Science and Technology. I'm Kathy O'Connor with Global Genes, and I'll be helping with this breakout session. As a reminder, we are live streaming this through Platform Q, so we will have some time for questions at the end, and make sure you use the microphone so everyone can hear you. So welcome to those remote attendees. The next session is Gene Sequencing, Not Just for Diagnosis, presented by Dr. Tamar Grossman, Associate Director at Ionis Pharmaceuticals, and Luke Hickey, the Senior Director of Human Biomedical Sciences at Pacific Biosciences. Please join me in welcoming them.
Great, thank you. Okay, well, I want to start by thanking the organizers. This is a very interesting conference so far. I'm actually from the technology development side, so I've spent about 16 years working on different sequencing platforms and for companies that actually specialize in developing genetic analysis tools. So we don't often get to see and interact with a lot of the patient communities. We're usually talking to the MDs and the clinical research folks. So it's a real treat for me to see actually how it helps impact the patient communities. So I wanted to thank the organizers for putting this together and then inviting us to participate. And I'll just start by saying, I think there's been a lot of talk at the conference about how to use genetic sequencing technologies and the benefit it can provide, both at an individual genetic disease level and then also for the diagnosis side. The talk today will mostly focus on sequencing of individual genes, and really how do you think about that? What potential advantage can it provide? How do the research communities think about doing this in a way that's beneficial to advance the science as they try to understand the genetics and what's happening? But I wanted to start just at a high level. I wasn't sure from an audience background what the experience level was with sequencing. Usually the experience with sequencing for most of this community will probably be interacting at the clinical level with some sort of a blood draw. And then we and I specialize in this part, which is development of these sequencing instruments that actually do the genetic analysis work. And we are now on the third generation of the technology. So the first iteration was a method called Sanger sequencing. So some of you for single gene testing may or may not have had some Sanger sequencing work done. The technology that most people are using these days for the whole exome sequencing and whole genome sequencing is the Illumina methodology with the sequencing by synthesis. And then we have actually developed this third generation technology called single molecule real-time sequencing. And I think the first speaker did a really nice job of describing the differences with some of the technologies. But if you look at read across the DNA or RNA, and I'll describe a little bit of how you can do that in the context of the disease. One of the questions asked by these technologies is, what is the read length? And the hallmark of the third generation technology is that the average read length is about 10,000 bases at a time. So you get a contiguous read of 10,000 base pairs, and that can actually be very informative for a lot of the different types of variants that are not currently as well served with some of the shorter read methodologies. So I think we heard about structural variation, and I'll provide some examples. So if your particular gene is known to have structural variants involved, then single gene sequencing with a maybe Sanger sequencing or third generation sequencing might be potentially more beneficial. And just to start at the high level, so I think we heard this morning about the human genome. This was published, and really when we talk about a human genome and we talk about 3 billion or 3.2 billion base pairs, it's a little bit of a misnomer. We actually have one copy of the genome from your mother, one copy from your father. So for every single gene, you actually have 2 copies. Either one of those copies can have a mutation that leads to deleterious results and potentially a negative phenotype. The genetic counselors will describe homozygous versus heterozygous mutations. Homozygous means the same on both alleles. Heterozygous means one genotype on one allele is different than the other. So those are terminology that you may or may not hear in the context of doing some research work into a single gene disorder. But the human genome, it's organized into 3 billion base pairs. It's 23 chromosomes. So if you get karyotypes done, or if you look at some of the cytogenetic array-based diagnostic methods, sometimes they'll give you something called a molecular karyotype. And it really is just an organization of chromosomes, and they show you genetic variation on individual chromosomes. Again, the genome encodes something called genes. and each gene encodes something called proteins. And the director of the U.S. National Institute of Health, Dr. Francis Collins, he does talk about the fact of trying to find heritability, so trying to find the underlying genetic cause of all of these different disorders really benefits from looking at genetics and studying DNA in a more comprehensive way. So we talked a little bit about the gene coding portion of the genome, the non-coding portion of the genome. The NIH are investing heavily in all of these areas to try to build even better maps of human genomes. And when we think about doing single gene sequencing, really what we're trying to get at is an understanding of how life is encoded. And it starts with the DNA, so it does start with the 3.2 billion bases. It starts with a particular gene. That gene is transcribed into a molecule called RNA, and this is a single-stranded molecule. It's a 4-base pair, or 4-base, not pair, encoding mechanism. And then those are actually read out into something called amino acids that actually form the proteins. And the proteins are the functional unit that actually does something. And if the code is incorrect, if you have a variant that is deleterious, if you don't have enough of a particular protein, you can get some of the clinical manifestations of disease, and those end up manifesting in different disease phenotypes. So really, when you talk about genetics research, what they're trying to do is understand how are variation in the DNA affecting the RNA, and then how are those changes in the RNA ultimately impacting the protein, and what does that look like? So the genetics research scientists, they're trying to figure this part out. They're trying to understand for any given disorder what this looks like. And genetic variations, I think we heard that there are simply differences in the genetic sequence between an individual and the human reference genome. The human reference genome is a little bit of a misnomer because it doesn't represent any single individual's genome really well. But there are variants. We don't always know what the variants are, and that's where the research comes in. But they can be in the genome and the DNA, they can be in the genes, they can be in the proteins and in the function of the proteins. So they're seen really at any of the different genetic levels. Variation is frequent across the genome, so we heard a nice summary of all of the different types of variants you can expect to find if you sequence a whole human genome. The same is actually true with genes as well. You'll actually find a lot of mutations, some of which are not bad. They just occur at random across the population. Sometimes they don't lead to disease. Sometimes you'll hear the term non-synonymous coding change, and it's just because if you look at the protein, there's actually lots of different ways to spell a single amino acid, and if the mutation doesn't change the actual amino acid, then sometimes the protein is fine, even though you have a variant. So that's something to know and think about as well. And a lot of the variation is harmful, unfortunately, and it can lead to genetic disease and genetic disorders. And when we try to understand at an individual gene level what the variation is, that's really some of the questions that we're asking is what is the variation that's occurring in the disease population? How does that variation actually impact some of the clinical phenotypes that we're seeing? And that opens the door to a lot of the downstream research work. And that's the goal of the human genetics. So this is just a map showing that if you did look at variation across global populations, and there's the 1,000 Genomes Project that is starting to do this and has been doing this, you will see a lot of normal differences in the human genome. So this is just some background information on normal human genetic background. So in terms of how sequencing is used to study disease, I won't spend much time on this because I think we've heard a lot about this morning. But you can do, in addition to the whole exome and whole genome sequencing, you can do this third part, which is high-resolution targeted sequencing of a specific gene or region of the genome. And if you're fortunate enough to know what the particular gene is, if you've discovered that for your rare disease, you can then design very targeted assays that look at exquisite detail at the variation within that particular gene. So that's something that technology is available now, and there are researchers that have particular interest in that. And what they're trying to do when they do study these individual genes, they're asking questions about genetic mechanisms underlying the disease. So we talked about how life is encoded, and really what people want to understand is, well, as you look at a specific mutation, how does that manifest in changes in the RNA? Does the RNA, is it put together differently? There's exons, introns that are spliced together. Does that splicing look different? Is the RNA encoding a different form of the protein? So they start asking very specific questions at that level when you get into the single gene sequencing. There's also regulations. So there's a field called epigenetics that also regulates the expression level within a gene, and that's something else that, from a single gene perspective, a lot of the researchers look at. And all of this underlying research work to understand a mechanism actually opens the door to developing different opportunities for biomarkers to classify patients within different subsets of the disease. We heard this morning the nice talk where they described how autism is actually an autism spectrum disorder. There's a lot of different genetic components that are involved with autism. And as you develop drugs, as you look at developing ways to segment the patient population, it's important to understand at the basic genetic level what the background is so that you can develop effective strategies that look at the genetic components of the disease, and you can actually focus in on the specific disease mechanism based on the genetic background. And that's really what this is discussing as well. So once you understand the disease mechanism, it opens up a lot of opportunities to make further progress downstream with thinking through those biomarker strategies or thinking through opportunities for a lot of the therapy development and newer research work into some of the genetic modification methods that we heard about this morning as well. And I'll just point this study out. So this was a nice study that is disease-causing gene discovery work. So this was done in the background of familial ALS and frontotemporal dementia, but there are a whole class of disorders that are caused not by single base changes, but actually by expansions of something called repeats. So there's a lot of different variation that occurs in the genome, some of which is repeat expansion. And I wanted to show just a couple of case studies how researchers are working on specific disease areas that are affected by these specific type of genetic changes called repeat expansion disorders. And there's a nice article that was put out by Ewan Ashley very recently in Nature Reviews Genetics, and he describes quite a few different disorders that are caused by repeat expansions. Huntington's disease is here, myotonic dystrophy, Fragile X syndrome. So there's quite a few different diseases that are caused by these repeat expansions. And where they manifest is the normal version of a gene may have a certain number of what we call repeats. In the case of Fragile X syndrome, this is the letter C followed by 2 letter Gs. And in normal individuals, they would have between 5 to 44 copies of that. If you have a genotype where you actually have an expansion, so that repeat is actually expanded, it's copied in more times into the genome, you end up developing into these what they call pre-mutation genotype, and then if you get above 200 copies, it ends up being a pathologic genotype. So this is a particular genetic variant that underlies Fragile X syndrome, and there are researchers that are studying this very actively, including Dr. Paul Hagerman and his wife, Rhonda Hagerman, up at the UC Davis Mind Institute. So we were fortunate enough to do a lot of collaborative work with Dr. Paul Hagerman. And really some of the research goals that they had were to understand a little bit more, because they know the genotypes that lead to Fragile X, but they don't have a very strong grasp of the disease mechanism. So they have an interest in sequencing the gene, even though you know the genotypes, sequencing the gene to help understand why are these repeat expansions happening, what happens if there's an interruption sequence, if there's an A interspersed with those CGG repeats, how is methylation related to the disease, and then can you develop some earlier screening methodologies? So in the state of California, we do have newborn screening. Can you develop strategies with newer technologies to actually do newborn screening to identify patients that are susceptible to developing Fragile X disease earlier? And then, of course, with earlier diagnosis, you can actually come up with intervention strategies to start earlier that also have a strong impact. So these are some of the clinical researchers that are doing disease-specific, gene-specific research work using the technology. And the mechanism in Fragile X disease, the typical genotype, as I mentioned, is about 45 copies of this repeat or less. And if you have that genotype, you have a normal amount of mRNA, so this intermediary, and then you have a normal amount of the FMRP protein. If you have a pre-mutation, so if you're in the 55 to 200 copy range, you actually have an increase in the amount of mRNA, and you have different disorders that are late onset adult disorders that are related to this. And if you have a full mutation, so greater than 200 copies of the gene, you actually get a phenomena that silences expression of the protein, and then you end up developing Fragile X syndrome due to the lack of the protein. So this is something that is described as the disease mechanism. And what researchers really want to understand is what does this mechanism look like, so they can start to come up with better strategies for hopefully developing some therapies or mechanisms to help improve some of the symptoms. So we've done some work. We have a very long-read sequencing platform, so we've done some targeted work with Dr. Hagerman to look at both genotyping these expansions at the single gene level using targeted methods, both using something called PCR to amplify, and then a newer non-amplification based method that preserves some of the mosaicism that we heard about. So there are newer technologies that you could use to look at these types of questions. He's also interested in studying the interspersion of these AGG interruptions. And again, these interruptions also have a clinical impact on anticipation. So the passage of an expanded allele to offspring, and that's something that they've been able to study as well. And I'll skip through that, but that's just an example. And then again, I think the goal is to understand the disease mechanism, and once you understand the disease mechanism, you have more opportunities to partner with therapy developers or understand how you would go about coming up with better diagnoses and better stratification methods, and then ultimately better treatment opportunities. And then I mentioned the RNA component as well. So because the pre-mutation carriers, it's a late onset adult disorder, but they still don't really have an idea as to, they know the RNA is overexpressed, but they don't necessarily understand what the disease mechanism is. So we've been working with Dr. Flora Tassoni, also at the UC Davis Mind Institute, to understand alternative splicing. And there is a mechanism that is related to alternative splicing and overexpression of certain isoforms that results potentially in some toxic mRNA that is hypothesized to be one of the disease mechanisms. So they've started to ask very specific questions about expression of individual isoforms, and then as you ask those questions in the context of a disease cohort, you can actually then identify how a clinical phenotype of a disease is correlated back to presence of these individual isoforms, and then think about developing mechanisms for treatment again. So this is really the foundational work that allows some of the downstream drug development process to happen, and a lot of the biomarker development process to happen as well. And then I'll just give one more example. So ataxias are another group of disorders that we've been fortunate to partner with some very active researchers on, and they're clinically and genetically heterogeneous group of disorders. So there's a lot of genetic causes. There's a lot of different genes that can lead to the clinical description of an ataxia, and they have different clinical descriptions. So if you go into, for example, Dr. John Day's clinic at Stanford School of Medicine, they will then try to understand at the clinical level what the phenotype is, and then based on that clinical observation, do some specific genetic testing, either single gene testing and or whole genome sequencing, and try to understand what the genetic components are. But ataxias are very similar in terms of the clinical presentation, but they do have a lot of different genetic individual genes and variant types that contribute. Most of the variant types are actually due to these repeat expansions again, so this is another example of repeat expansions being the cause of these diseases. And in the case of the work we've been doing it on SCA10, so type 10, and it's caused by large non-coding ATT-CT repeat expansion. And again, this is an example of a non-coding variant, so this is not occurring in the protein coding exome. So it's a variant type that you would have to look either at the whole genome to see or use other different methodologies to be able to detect the variant type. And in the case of SCA10, the pathogenic alleles, they range from 280 to 4,500 repeats. So this is a lot of bases that we're talking about being affected. It's a repeat expansion disease. So they've been studying it in the case of that. And one of the important things with these disorders is that the interruption motifs, they actually act as the phenotypic modifiers. And what that means is that instead of just seeing ATT-CT repeated between 280 to 4,500 times, sometimes there's interruptions. You have this repeat sequence, but then bases in between that look different. So Dr. Tetsu Ashizawa from Houston Methodist Research Institute, his lab is focused on these ataxias. They've been studying these at the clinical level, trying to understand how some of these interruptions are correlated back to different phenotypes that they see in the clinic. And they had some nice work that they published where they show different interruption sequence associated with different clinical presentations. And this really forms the background for doing some of the disease patient stratification work that we talked about before, and then understanding both from a biomarker strategy, how do you develop biomarkers to bin individuals, and then of course downstream from a drug discovery perspective that gives companies targets to work with at that point as well. And again, the hypothesized disease mechanism is through this RNA transcripts, and in this case, they inhibit critical RNA binding proteins, resulting in loss of function. So this disease mechanism also gives you a good sense of how the disease, based on the variant, how it's progressing through the RNA, what the impact is on the protein, and it opens up a lot of doors downstream for researchers to work with. And I'll skip through some of these. The paper has been published. There are some interesting conclusions that they drew based on that. And I think one of the nice things is the ability to use these types of approaches to look at disorders. Once you do have a gene that you know is involved with the disease, you can implement these types of strategies to do very detailed genetic work at the DNA level, at the RNA level, at the protein level to understand the disease mechanism, and that really sets the stage for different research discoveries downstream. So hopefully that gives you a good perspective of the way in which researchers are using these methods for single gene sequencing, and really, after you have a diagnosis, why is it important to study the gene further and understand the genetics? Well, this is the reason. And just to conclude, if you do sequence the genes, it gives you a lot of understanding into the genetic basis of the disease. You can understand the heterogeneity. So how, if you have 100 patients that have the disorder, sometimes they don't all have the same exact symptoms. How are they different at the genetic level? That helps you understand what the mechanism is. And as you explore the relationship between the DNA and the RNA and the protein, it does give that good background on the disease mechanism. The researchers typically publish this work once they do it, and that opens it up for other researchers at other clinics to also make advances in the disease area. So this is into the information sharing, so it's not locked into one lab or one specialist side. And then the new methods are coming out every day. Being in the technology sector for the past 16 years, I am always amazed by the advances that are being made, and I think that's a benefit of a lot of the research funding that happens. So advocate for research funding, not only at the NIH level from the government, but within individual disease advocacy groups as well. And I think we heard from Autism Speaks this morning, but those are phenomenal sources of funding for researchers that are interested in studying disease at the single gene level. And then I'll also say that these scientists, so the Dr. Hagermans of the world, the Dr. Ashizawas of the world, they really rely on an active and involved patient community. So I think to speak on their behalf, I think one of the things they would encourage is for the community to get interested in genetics, get interested in participating in, if they are enrolling in genetic research studies, really engage with them. I think these clinicians you'll find are very passionate, they're very involved, eager to work with the community. I know Dr. Hagerman goes to the Fragile X conference every year, and they're very passionate about this area. So I would encourage you to get involved. Hopefully I provided some background as to what the technology, what doors that has opened up in terms of genetic research work. And I think I'll hand it over to my co-speaker for the session here with Tamar. Thank you.
Hello, everyone. I'd like to start by thanking Global Genes for inviting me to give this presentation. I work at Ionis Pharmaceuticals, and I'm at the drug discovery, and what that means is that my daily job is to be on the quest for finding cures to all these rare genetic diseases. So being here means a lot to me, and it's very important to meet with the patient advocacy, and learning more about disease, and mostly getting inspired by your work. So today I'm planning to tell you what is beyond the diagnostic. You have a diagnostic, you know what your child had, but most important is how do you find a cure for the disease, and what do we do using genomic data to find cures for disease, and how we take that forward. So I'll start by giving you some introduction for gene sequencing in drug discovery and giving you an example where we use the genomic sequencing. I'll give you an introduction to our technology because that's key, and a unique technology to target the genes. Basically, we target the RNA. And I'll give some examples for diseases in which we develop therapies, and the drug discovery was based on genomics findings. So we've heard today of how we can use and sequence patients. Once they're diagnosed, once we have a patient population, sequencing the patients to find new molecular basis of the disease, understanding the mechanisms, but also finding novel targets for therapeutics. Basically, we start with patient recruitment. The next step is collecting the samples, could be blood, tissue, cells, and doing RNA or DNA sequencing to identify the genes that are changed between the normals and the disease, and using all kinds of omics software to learn more about the pathways. And that leads us to finding disease gene, modifier genes that helps us design new targets and new drugs, and finding biomarkers for the diseases. So with the first step, we identified disease-causing mutations, and we heard a lot about that today. And the mutations could span many different types. Some of them could be a loss of function, where you just completely lost the function of a protein, or you can have a gain of function, where you have too much activity of the protein. We also have splicing mutation, which we touched upon earlier, and these are just mis-editing of the messenger RNA that forms a defected protein, and I'll give examples for that. So this is what sequencing of the patients will give us, but we can also use sequencing to predict the response to specific drugs. We can determine if there's a drug resistant in certain patient population. We can take responders and non-responders and learn more about the genes that change and are contributing to that. And we can also learn about modifier genes in individuals that have the disease genes but don't present the disease. So that can help us also find new targets for therapeutics. In addition, we spend effort in finding disease progression biomarkers, which tell us on how the disease progression and the severity of the disease. We're identifying targets, but also having information that helps us design clinical trials and monitor the patients during the trial to see the efficacy of the drug. So a couple words on the Antisense Technology. I'll try to make sense into this antisense. We're targeting RNA. And we believe that RNA therapeutics is really revolutionary because you're targeting basically the gene that is defective, or genes that are affecting the disease. And it's a class of molecules, and we learned about it today, that are responsible for regulating, making the protein, and are important for many different diseases. And by targeting RNA, we can not only target the protein-making RNAs, but we also heard today about the non-coding RNAs. Some of them are related to disease, and with the RNA antisense technology, we can target them. So we saw the dogma of where we have, for example, a mutant gene that makes a messenger RNA that's been translated and making a protein with a mutation, a defected protein that's causing a disease. And what our antisense does is binding the messenger RNA, and its a messenger RNA is a single strain, the antisense is another single strain. They form a duplex, and what happens when they bind is messenger RNA is getting degraded. And by degrading the mutant or the defected messenger RNA, you do not form the mutated protein, and you cure or stop progression of disease. So this is the antisense drugs. They're basically a piece of DNA. This is the drug. So the DNA is formed by a core piece of DNA of the drug that is getting recognized and getting degraded once it binds the messenger RNA. But it also has some chemical modifications on the ends of these piece of DNA, which are contributing to the affinity to the target messenger RNA, the stability of those drugs in the system, and also the tolerability. Our drugs has been in over thousands of patients from many years and different doses, and they've been very safe. So We're using the antisense for multiple mechanisms. And these are some examples from drugs that we have in the pipeline in which those antisense drugs are targeting different mechanisms of disease. For example, in the TTR amyloidosis that is in phase 3 trial now, we have a drug that is targeting and reducing the target RNA that has the mutation, the defected protein, and helping to remove those defected protein to cure the disease. In the myotonic dystrophy 1, which is the most common muscle dystrophy, our drug is targeting a toxic RNA. So in this disease, it's a non-coding RNA that's forming a DMPK RNA that also has the repeats that we heard earlier about. And these antisense is binding this defected repeat RNA and removing it and helping to remove the pathological aspect of the disease. Another aspect of our mechanism is shown here, and this is with a drug for spinal muscular atrophy. In this drug, we are increasing the production of therapeutic protein. So this drug is different because it's changing the splicing of the protein. So the editing of those messenger RNA molecule to correct and make a normal therapeutic protein. And this is in phase 3 trials, our most advanced drug, and it's really helping and saving lives of children with spinal muscular atrophy. So these were some examples of the mechanism of the antisense drugs, but now I want to focus on some examples for where we used genetic information to design specific drugs that are unique to the disease. So the first example is retinitis pigmentosa. This is a disease that causes blindness. It's an inherited disease, and it results in the loss of the photoreceptors. It's gradually progressing, and it's affecting children. And as you can see here, this is the retina of this thing. The black cells here are the cells that are dying because of the mutation. The children and the patients that are affected have a gun barrel visual field, so basically they only see this much of a visual field. And blindness occurs in middle ages. The disease is caused by a mutation in rhodopsin gene. Rhodopsin is a very important protein that is important for our vision, and it is a rare disease population. What's tricky about this disease is that rhodopsin is very critical for our vision. So we don't want to target the normal rhodopsin gene. We want to target only the mutant, only the defected one. And since this disease is an autosomal dominant, that means that every patient have a normal rhodopsin and a defected one. So we want to make sure that we're targeting only the mutant one. And this is what we are planning for. This is P23H. These dots represent those amino acids, and this is showing the structure of this rhodopsin, very large transmembrane protein, and this circle here is the mutation that we're targeting. So we're only targeting 1 point mutation with our oligos, and since we have a DNA that could be modified and designed specifically to mutation, we can use the RNA Antisense Technology to do so. So this is some of the data. I'll just quickly summarize it. It's basically showing that when we design it, we've screened for many oligos, and we identify specific oligos that can reduce the mutant allele without touching the normal allele, and this is shown here. So this is giving increased concentration of the drug, and this is in cell lines. You can see that the human normal wild-type rhodopsin is untouched, where there's a dose-dependent reduction of this mutant. And this was also repeated in humanized mice. Mouse models are critical for our proof of concept study before we move forward our drugs, and we were able to show that in these mouse models that express the normal and the mutant, we're able to only target the mutant. So hopefully this novel therapeutic strategy could be helpful and restore vision in children with retinitis pigmentosa. And this is at very early stages, preclinical stage. And moving on to the next example, this is another disease. It's called adult polyglucosan body disease. And in this is a late onset disease. It starts affecting individuals at their 40s, and it's a slow progressive disease that is affecting the brain and the muscle and patients have many different problems that is progressive and end up in a wheelchair with uncontrolled bladder, problems with gaits, and also dementia. The pathological finding of this disease is those, as you can see here, it's a slice of the brain, and you can see those dots here. This is a stain for glycogen. So this is basically a glycogen storage disease where you have accumulation of those polyglucosan bodies. And they're found in peripheral tissue, all over in the muscle, and also in the brain. And the reason these patients have those accumulation of is because they have mutations in the branching enzyme. So usually glycogen, which is the storage of glucose energy in our body, is stored as a spheric molecule that is highly soluble, and it's formed by glycogen synthase, which synthesizes the glycogen, and the branching enzyme that condenses it and making those soluble molecules. When you have mutations in the GBE or the branching enzyme, the glycogen is getting elongated and forming those fibers, and then they are insoluble and making all these, what you showed here, it's the polyglucosan aggregates, which are putting a significant stress on the neurons and ending up in neuronal death. So the problem in this disease is that you have a misregulation of formation of glycogen, which forms those polyglucosan bodies. This is a founder mutation in Ashkenazi Jews. There's also other mutations. And the story here is when I worked with the Foundation for APBD, and I talked to one of the investigators, he told me that he recently found a novel mutation. And the story is that these patients did not have the common mutation that most of the Ashkenazi Jews have, and they have a very severe pathology. And when he went ahead and sequenced the gene, and he sequenced the DNA and the RNA, he found an intronic mutation. So we talked about this intron, so it can be junk DNA that we get rid of, but this junk DNA can sometimes possess some really critical region. And when you have mutations in it, you can also have disease, and we've heard a couple of examples of that. And what we found and this is the sequencing that is done in the patient, is that in the individuals, and he had 35 patients that he sequenced, he found an additional sequence that ended up inside these normal genes. And what happened is this editing mechanism to get rid of the intron didn't work well. So the mutation was basically here in between, in the intron. So the normal genes have exons and introns in between, and then getting spliced out and edited, and you end up with a messenger RNA that contains no introns. But what happens with the mutation in that particular patient population is that there's a pseudo-exon that's getting incorporated into this messenger RNA that's not supposed to be there, and that forms a defected protein and causes the disease. So when I talked to the Orhan Akman from Colombia, who asked, can we do something for the patient? I told him, well, let's try, and let's try to target these sequence that is within the intron and see if we can stop that abnormal inclusion of this intron. And this is what we did. We screened for many oligos, and we were able to find, this is just showing the increase in the normal GBE gene. So basically what this is showing, that we're able to correct the mutation there and increase the levels of the normal gene. And not only that we increased the messenger RNA level, we could test the protein activity. And when we looked at the activity, we were able to also increase the activity. So this is kind of a personalized drug that will work for this small population of patients that will be able to increase and improve their activity. And this is just showing in another study that we did with patient fibroblasts, and we're able to increase and correct the activity of this enzyme. So potentially this could provide help and much needed therapy for this progressive disease. The last example that I have is one of our most advanced programs, and this is for the ApoC3. And the rationale for that came from its function in triglyceride mechanism. But a strong genomic validation came from studies in the Amish population, and it was called the Amish protective gene, because this was found to be mutated in this population. So these studies took over 75,000 individuals and sequenced their genomes, and what they found is that all these patients that have reduced levels of triglycerides also had reduced or knockdown level or non-existent level of ApoC3. So these actually show that loss of function or elimination mutations in the ApoC3 gene is associated with very low triglyceride and very low risk of cardiovascular disease, validating it as a target for triglyceride-related elevated disorders. So the disease that we've decided to focus on for our clinical trial is the familial chylomicronemia syndrome, or FCS. And this disease is a rare disorder, and there are loss of function in genes that are responsible for triglyceride mechanism. So there are mutations in the LPL, which is the lipase that degrade the triglycerides, and when you don't have that, you have very high level of triglycerides. And in these patients, in the thousands. So the patients suffer from very, very high triglyceride, and there's no drug for them, no treatment, except for a very restrictive diet that contains no fat. So that's very, very challenging to do. But if they consume even a little amount of fat, these patients have a very severe elevation of chylomicrons and triglycerides, and they suffer from severe hepatitis, abdominal pain, and many other complications. And there was nothing out there for these patients. And they also have what's called the xanthomas. These are because they have such a high level of the triglyceride, they actually erupt in tumor-like skin lesions on their skin. So this is another characteristic of the disease. A simple blood test could detect this disease. And as you can see here, it's an example. This is the normal plasma, and this is a plasma for M patients. So you can see the plasma is really milky. This is due to the high level of the triglyceride, which was a very high rate of cardiovascular disease and also has other issues, as I mentioned. So we have this drug, it's in phase 3, and I'm showing some data that was published in the New England Journal of Medicine. Patients that were treated with the drug showed a dramatic drop in their ApoC3 level, 90% knockdown of this gene. In addition to that, they have 86% reduction in their triglyceride, and the levels of this patient that was in the 1,000 to start with went down below 500, which is close to the normal level of the triglyceride. So this is very exciting, and this is in phase 3 clinical trial and also in other indications, and we hope that this will be approved soon and get to the patient as soon as possible. So I'll stop here and just summarize, and we've seen by Dr. Ring a very nice presentation of the drug discovery. This is more or less the drug discovery path for a process for our antisense drug and where we use the genomic data. So most of the genomic data and the efforts are being used in exploratory research where we identify the target, use the proof of concept studies in the animal models and identify ways to validate the target. We next move to the drug discovery of the human candidate, and we do the screens and we do the humanized mouse to find the best candidate for humans. We move on to do the preclinical development and the IND-enabling tox study, and we initiate natural history studies, which are really critical for some of those rare diseases when there are very few patients and not a lot of know is the progression of the disease, so this is critical too at this stage. And we end up with a clinical development or first in man, that in rare disease is usually a patient, and we determine the tox and do other chronic tox studies and get hopefully to marketing and getting the drug to the patient. And at this stage, we use biomarkers and do patient response studies to learn more about the drug and the efficacy. So I'll end here, and I hope I showed you that we can take this information from genomics beyond diagnostic to do a well-educated and further research to identify gene target and use the RNA technology, which seemed to be, to me, a straightforward way to try and target the drugs and find therapeutics. I'll be happy to take questions now.
Thank you so much, Dr. Grossman and Luke, for diving deeper into the different uses and benefits of sequencing. We do have about 5 more minutes before we break for lunch, so if there are any questions, I will be happy to bring the mic to you.
This question is for Luke. What is your detector in the SMRT method?
I'm sorry, do you mind repeating the end part of that?
Your detector?
The detector. Yeah, so the technology is, it's interesting. It's not necessarily consequential to the way that we do it, but we look at a single molecule. It's a fluorescent signal that we look at, and we actually label each nucleotide with a fluorophore, so 4 different colors, similar to the Sanger sequencing process. And then as the nucleotide is added, it's actually held in the detection chamber, and then we observe the light, and that actually gives us the base call. So we have more details. It's published online, but that's the detection.
So it's a UV detector or photo detector?
It's actually a camera, so I believe it's actually a CCD camera. We take movies as opposed to single still images. Yeah.
Okay, thank you.
Anyone else?
How do you make sure that the RNA sequence that you're using in the oligo doesn't match some other sequence someplace else in the 3.2 million base pairs? And how do you make sure that that doesn't happen?
When we design our RNA, this is a very good question. We make sure that the sequence is unique and doesn't have any of the off-target, what we call, for targeting other genes. And we also have capabilities to do RNA-seq and also kind of validate that by treating in the cells or in the mouse models.
I have a question for Dr. Grossman about the antisense. Is this only appropriate for gain-of-function mutations? Because I would think if you had a disease where the mechanism was a loss of function of the protein leads to disease, and your therapy is binding the RNA and degrading the protein, that you're not fixing it necessarily, you're getting rid of the protein. So how does that work?
We have drugs for gain-of-function that are specific for gain-of-function mutation. These are like the low-hanging fruit. You have too much of a protein, you want to reduce it. But our other drug, like for example, ApoC3, is another enzyme in the pathway. It's not the disease gene. The disease gene is LPL, but we have we're targeting somewhere else in the pathway to correct the pathology. We also have and recently published a paper on actually upregulation of some of the targets by targeting other sequences, so
Okay,
using
great.
the oligos. So we have a broad range of mechanisms that we can use.
This is probably a silly question, but are the drugs administered systemically or on organ systems?
That's a good question. For our CNS drugs, we administer intrathecally. For children, the half-life is very long, so the children, for example, with SMA are getting once or twice a year injections into their spinal cord. For our other drugs that are targeting systemic drugs, the drug is administered by subcutaneous injection once a week.
Anybody else? Okay, thank you again so much for presenting today. I did want to tell you that it's time for lunch. The lunch will be held out on the other side of the main ballroom on the grass. The hotel has asked that when we exit this room, we turn right instead of going back around. Someone else is having a lunch in that corridor. So if you go out right here, turn right again, and then you'll see the corridor down to the main ballroom. So thank you again so much for your time today.